Hello everyone, and thanks to the organizer to give me the opportunity to present a work about the study of the catalytic cycle of human transketolase and to draw some conclusion. During this presentation, I wish to highlight the contribution of molecular modeling on the knowledge of chemical or biochemical reaction pathway. During the submission of articles, I was able to measure the lack of credibility of experimental research toward the theoretical proposal. First of all, a presentation of the study framework. Transketolase is an enzyme of the non-oxidative pentose phosphate pathway and is an homodimer of 20,700 atoms. The initial objective was to use this enzyme in the synthesis of new molecules. Here is a commonly accepted reaction mechanism of transketolase, or TK. The enzyme ensures the catalytic transfer of hydroxyl acetyl groups between a donor substrate, ketose, and an acceptor substrate, analdose. This mechanism requires the presence of a cofactor, thiamine diphosphate, or THDP, which must be activated for the reaction to take place. In 1951, Mizuhara showed that thiamine in water gives the same results as aldolization. In 1958, Breslau described the mechanism of aldolization catalyzed by the thiamine in two steps. In 1992, the first crystallographic structure of TK is published, but without water molecules. In 1993, the V form of THDP is defined as the biologically active form by comparison of crystallographic structure of different THDP-dependent enzymes. This slide shows the scheme of the THDP activation mechanism proposed since the publication of the 1-TRCAP crystallographic structure. This activation mechanism comprises 5 steps and no water molecule. The key step is the activation of C2H by the alpha-iminium group of THDP. This is a self-activation model. However, we can see 3 chemical inconsistencies. As type C, the histidine is not correctly oriented, and the pKa rules are not respected between an aromatic iminium and an histidine, in this case, which other bases. At type D, the intramolecular proton transfer is unaccountable with the 75 degree angle between the anti-bonding molecular orbital of the CH bond and the non-bonding electron pair of the nitrogen. Finally, the pKa of the proton at C2 position is between 6 and 70 to 19, depending on the references. To answer the question, how is the THDP activated, we use onium 2-layer, a QMMM method, mixed method, quantum mechanics, classical mechanics, which make it possible to study a reaction pathway on very large systems. This single slide summarizes a third of the work time. Indeed, starting from the 3 MOS, there are 3 important steps. The first one is to find the right ionization state of the histidine, or if they are protonated in N or N delta N epsilon or N delta position. In our case, ProPK did not give good results because it could not take into account the acidity of THDP. The second step is to position correctly the water molecules and to define correctly all hydrogen bonds between the water molecule, the amino acid, and the cofactor. An error of these two first steps makes that the results after minimization diverge from the crystallographic structure. Many tests have been done before finding the right solution. The initial system was too big, so we decided to reduce the size of the system to a sphere of 20 angstrom radius, whose center was the C2 of THDP. But we had to fix the position of the heavy atoms peripheral to our system, otherwise we would lose the structure after minimization. Our theoretical biochemical model for the QMMM study include a total of 5,156 atoms. Finally, the last step is to define the quantum subsystem or QM part, such that the quantum subsystem during the unconstrained minimization on the high layer, the crystallographic structure of this high layer is conserved. Again, the process is iterative. We had to include backbone atom far enough from the reaction site to ensure the coherence of the QM part. So we have 4,979 atoms in a classical part and 164 atoms in a quantum part. In the quantum part, all atoms are free. In the classical part, all hydrogen atoms and some heavy atoms are free. Let's take a quick look at the procedure followed. The initial state present here is the result of the procedure present in the previous slide and is perfectly superposable to the three-most crystallographic structure. First, from the initial state, we scan on the presumed reaction coordinate. Here, the elongation of the CH bound. From this first results, we look for the transition state close to the highest energy point of scan. 3. The transition state is validated by an intrinsic reaction coordinate procedure, which must lead to the reactant and the product of the study reaction. Finally, the structure resulting from the ERC are minimized. The Gaussian command line is described here, and you may notice that we use the 331G basis set, which may seem insufficient for modelers, but I will come back to this at the end. Here is the result of our calculation on the right side compared to the activation mechanism previously described in the literature to the left side. 4 fundamental differences can be observed. The activation mechanism is done in only one step, instead of 5 steps. The final acceptor of the proton at C2 position is the histidine 110A and not the imine at N4' position. A water molecule is essential to ensure the proton transfer to ensure the transfer of proton. The N4' iminium play a role, in stabilizing the carbanion formed. We therefore propose a new model of self-stabilization model instead of self-activation model. Here is the movie resulting from the calculation. We can notice that between the two states, the displacement of the atom and more specifically the hydrogen atom are very limited. From the energetic point of view, we can postulate that activation is spontaneous with dimerization. The Gibbs free energy of the TS is negative, and there is a continuous decrease in the Gibbs free energy between the inactivated state and the activated state. So no TS exists. The backward step is therefore impossible. Only the activated state exists in the TK and is very well stabilized with an energy of minus 69 kJ per mole. We have just seen the THDP activation. Now let's see the enzymatic reaction itself. On this slide, the complete reaction mechanism of TK proposed in 1998 by Schneider and Lindquist, circle in red, the point that we are going to see now. To study this first stage on the catalytic cycle, we start from the crystallographic structure 4KXV, in which xylulose 5-phosphate reacted with activated THDP. From this starting point, which is in fact a reaction intermediate of the first stage, we perform two scans, one leading to the reactant and the other to the product. The rest of the procedure is identical to the study of the THDP activation. Here you have the results of the addition of THDP activated on xylulose 5-phosphate. The two differences with the literature mechanism are, firstly, no base involved in the protonation of the form alcoholates. Secondly, the N4' iminium stabilizes the alcoholate by transferring the interaction with the carbanion to the alcoholate. Yeah. A short movie is better than a long speech. It is possible to visualize the displacement of the interaction of the N4' iminium group with the carbanion toward the alcoholate formed. The next difference is that the Breslow intermediate acyl or acyl enzyme is an enolate always stabilized by the N4' iminium group. This is in fact a continuation of the previous remark here. Here is a movie of the formation of the acyl enzyme and the release of glyceraldehyde 3-phosphate. On the right, the Gibbs free energy profile of this first stage. The blue curves represents this work and is compared to the calculation made on the current mechanism in the literature, red and magenta curves. Our energy profile is more favorable to the formation of the acyl enzyme. The limiting step is the addition of the activated THDP on the cellulose 5-phosphate, and this can be understood by the strong interaction between the carbanion and the N4-primiminium group, minus 69 kJ per mole. To study the second stage of the catalytic cycle, we start from the crystallographic structure CAT4KXX, in which cetoeptulose 7-phosphate reacted with the activated THDP. From this starting point, which is in fact a reaction intermediate of the second stage, we perform two scans, one leading to the reactant or to acyl enzyme, and the other to the product or to cetoeptulose release. The rest of the procedure is identical to the study of THDP activation. For the first step of this second stage, there is no differences with the current mechanism of the literature. Here is a movie of the reaction path of the addition of acyl enzyme on the ribose 5-phosphate. However, for the second step of the second stage, an important difference appears in the regeneration of the active enzyme. In the current literature model, the THDP return to inactivated CH2H form, whereas in our model, the THDP remain in the activated form, but stabilized by the N4-primiminium group. The THDP activation occurs only once at the dimerization of the enzyme and not at each catalytic cycle. This is more logical with the respect to the enzymatic kinetic. Here is the movie of the reaction pathway of this last step. On the left, the Gibbs free energy profile of this last second stage. This time, this is the second step, which is limiting. Here again, it is not surprising if we remember the stabilization energy of the alcoholate by the N4-primiminium group, which was minus 60 kJ per mole. At this stage, we transferred this interaction from the alcoholate to the carbanion. You can see here. This profile is also consistent with the advancement of the catalytic cycle. Let us now see why we use the C31G basis set, which was approached by the reviewer on the idea that the higher the basis set, the better the results. The following will prove that this postulate is false when we are talking about interaction between on-bond atoms. In the first study on the YES-T car, we made the calculation with the C31G DP basis set, which is considered to be more precise than the C31G basis set. You have here the comparison of the energy profile with the two basis sets. The other calculation parameters are strictly identical. On the left side, the onium energy in red is similar to the energy of the high layer in blue. The quantum subsystem does not create tension on the classical subsystem whose peripheral atoms of the backbone are fixed on their crystallographic position. We deduce that the cis-3,1G basis set is compatible with the crystallographic Cartesian coordinate. On the right side, the onium energy is in red is largely higher than the energy of the quantum part in blue. This is explained by the attention applied to the classical subsystem by the quantum subsystem. At the optimization of geometry, there is a dilation of the quantum subsystem, which imposes a constraint on the classical subsystem. Some atoms being frozen in the classical subsystem on their crystallographic position. This leads to an increased energy of the lower layer and therefore the onium energy. We deduce that the cis-3,1G DP basis set is not compatible with the crystallographic Cartesian coordinates. To conclude, we give priority to the definition of the chemical system over the size of basis set. To properly and accurately define the enzyme system, we have to check the ionization state of ionizable amino acid, beware of propagation if cofactor is present, the protonation of an N delta on R epsilon and epsilon position of histidine according to the possible hydrogens bond. The position of the water molecule and the justification of all hydrogen bond. Define the high layer so that it is stable without constraints. Do not hesitate to enlarge the high layer or quantum part. Use the cis-3,1G basis set, which is compatible with the crystallographic structure and allow to have a large quantum part. The cis-3,1G basis set allows to introduce easily a thermodynamic correction, which use the second derivatives. The latter are correctly defined because of being at the minimum energy of the basis set used. Let me thank my colleague and co-author, Lionel Noton, for crystallographic structure refinement and static structure, Laurent Sequier, and you for your attention. Thank you.